Now we are going to understand the salient features of the secondary growth. Now what is secondary growth? It is that process by virtue of which there is growth in the size, or rather I would say the width of stem and the root, and this occurs only in the dicot stems and dicot roots. You would not find the secondary growth occurring in monocot stem or a monocot root, with a few exceptions of course. In the case of monocot stems, we primarily find this secondary growth in the dicot stems and dicot roots. Now this entire process is responsible for increasing the width of the stem or the root, which of course, as I told you, would be dicot in nature. And what we are going to understand is the anatomical features and what changes are occurring inside the stem at which anatomical level. So when we understand the secondary growth in a dicot stem, we come across 4 different steps, and you are to be well versed with these 4 steps by the end of this lesson. Now the first step is that there is a formation of cambial ring. This is the first step. The second step happens to be that the cambial ring that is formed in the first step, that becomes active. Third step is, due to the activity of this particular cambial ring which is formed in the first step, there is formation of annual rings that takes place, and you must have observed a wooden log. There are concentric rings which are formed. It is those annual rings we are talking about. And last is the activity of cork cambium, and we are going to get introduced to few terms one by one as we go across these points. So the first thing that is that there is formation of a cambial ring. Now if you can recall the diagram that you made about the dicot stem, which gave its transverse section, we had seen that there was presence of cambium between the xylem and phloem. Okay, the vascular bundles were called open because there was presence of cambium in between them. If I have to represent it roughly, I would say that this upper part happened to be xylem, the lower one was upper part was phloem, lower one was xylem. In this way, in between we had a cambial ring, not ring, a patch of cambium, and this cambium is meristematic in nature. Now for understanding and for recalling what is meristem, what is the purpose of meristem, and how many types of meristematic tissues are present in the plants, that you are very well aware from the knowledge of your previous class. Again, I am going to just refresh your knowledge that there are 2 types of meristems, or 2 types of meristematic tissues. One is primary meristem and the second one is the secondary meristem. Now what is the difference between primary meristem and secondary meristem? First of all, you know what is a meristematic tissue. Meristematic tissue is that type of tissue which gives rise to new cells. Okay, it incessantly divides, it never loses its ability to divide, and new cells are formed. Now the meristem which is formed in the beginning from the seedling itself, that is known as primary meristem, and the secondary meristem is derived from those cells which are derived, or you can say formed, from the primary meristem. So these are, you can say, essence. In the beginning, the essence of the plant life lies in the primary meristem. The cells, new cells, are formed from the primary meristem. At some stage of the growth of plant, this primary meristem cells, at some stage of the plant life, the cells which had originated from the primary meristem had lost the ability to divide, can again become meristematic and form the secondary meristem. In case you are not able to relate to the essence of this meristem thing properly with reference to this topic, because I am just summing it short, you can watch the video where we have discussed the types of vascular tissues and specifically the meristematic tissues, the tissues in plant, that is a part of class 11. You can see that video and then you can relate in a better way to this topic. So coming back, we were talking that we had cambium present between the open vascular bundles of a dicot stem. This cambium is going to play the crucial role in the secondary growth. Now this secondary growth is going to be responsible for increase in the width of the stem. Okay, and later on, since your childhood, you know that the term for that particular stem growth is known as trunk, the tree trunk that we talk about. So this cambium inside the vascular bundles is primary meristem. First thing that you have to remember, that it is primary in origin. This is the primary meristem. It is primary in origin. How it is primary in the origin? Because in the beginning, when the xylem and phloem are formed, at that time only the cambium itself is formed. Later on, what will happen, that this cambium would become active and it would give rise to new cells. This is what is actual summing short of the secondary growth. But one by one, when we understand it, we first of all have to know that the cambium is the primary meristem which would give rise to new cells, and the cells which would be formed from this primary meristem would be secondary in nature. So, because this cambium lies between xylem and phloem, and this xylem and phloem initially present inside the vascular bundle are formed from the primary meristem, they are primary xylem and primary phloem. The cambium which is active now, the cells that it is adding would be secondary xylem and secondary phloem. By the end of the lesson, you will get to know how these are arranged, what is their purpose, how these are formed basically, that we have to talk about. So, we discussed that the cambial ring is formed. Now, as you can associate, or as you can see over here, we are talking about cambium in detail, but we have mentioned the main point of development in the secondary growth is the formation of a cambial ring, and cambium in no terms forms a ring. That's what we have studied. Now you can recall your knowledge from the lessons where we studied about the structure of a Dicotstem. I'm not going in depth because we have talked about it. Let's talk about these as the vascular bundles. This is what a Dicotstem looks like. Apart from the wavy endodermis and rest of the things that you have discussed, you don't get in detail of that. This particular region, as I am marking in red color, this is the cambium. These egg-like structures that I have drawn, they are the vascular bundles, and because I have to show them as open vascular bundles which are responsible for secondary growth, these open vascular bundles are having this patch of cambium. Alright, this cambium is primary meristem, meristematic tissue. These are the vascular bundles. Here we have xylem towards inside, xylem, xylem, X represents the xylem, and we have phloems. So, this forms the open conjoint vascular bundle in a Dicotstem. Now, this part, as you can see, it would form a ring in case this part is joined. Alright, this part. In case it is joined, then we can somehow arrive to the stage where we have formation of cambial ring. Now, this particular region is otherwise known as medullary ray. If you can recall, again I would say. Now, this medullary ray is parenchymatous. Again, repetition of the previous class material that we had done while we studied the sections of Dicotstem, where we had to understand how to represent it diagrammatically. We had talked about the medullary rays, the patch between the adjoining vascular bundles happen to be medullary rays. So, this is medullary ray, right? Now, what would happen in order to form the cambial ring? The cells which are parenchymatous and are in close proximity to the cambium which is present between the vascular bundles, they would become meristematic and there would be formation of a cambial ring. So, this process has to take place, and that we study as the first or the very first step in the secondary growth. So, we are arriving to somewhat thing that looks like a ring. Don't worry, we are going to have a projector presentation so that you can associate in a better way with good diagrams. Mine is just a rough representation. So, we get a cambial ring. Now, what is to be remembered over here before we pile up this first point where the cambium is formed, this particular cambium that lies between the vascular bundles, that is primary cambium. Okay, and this primary cambium, because it is present between the vascular bundles, it is known as interfascicular cambium. Oh, not inter, it's intra. Intrafascicular cambium. This is the first term that is to be remembered. Apart from the things which we are studying, they are to be remembered. Now you just have to cram this term, that the cambium that lies between the vascular bundles, that is known as intrafascicular cambium. The cambium which is formed by the transformation of parenchymatous cells in the medullary ray is the secondary cambium, because it has been formed not by the cells in the beginning but by de-differentiation of the parenchymatous cells, which were at some stage formed by the meristem, that was primary. So they form the secondary cambium, and the name for this cambium which is responsible for completing the ring is interfascicular cambium. Right. So we have two types of cambiums. One is intrafascicular, other is interfascicular. Because they are lying in continuity, they form a cambial ring. So with this, we come to the end of the first point, that is the formation of cambial ring, how it is formed. I hope you can associate this as the Dicotstem. Because I have not drawn, don't get confused, it would be having endodermis over here, the wavy endodermis, then there would be pericycle. Rest of the points are all similar to the Dicotstem that you drew and understood. Only thing why I have highlighted this part where we are talking about vascular bundles, because this is the region which would be responsible for the secondary growth. So the first point is done now, that how the cambial ring is formed. Cambium which was present initially in between the vascular bundles, that becomes activated, that is part of activity. But for now, what we are concerned about is how the region of medullary rays becomes meristematic and forms the cambial ring. We have intrafascicular cambium and interfascicular cambium, both together they form a cambial ring, and that is what we study in the first point. Now in the next lesson we are going to study the second point. It's better we cut short this lesson at this stage only, because in case we start studying it in detail, it would be a long lesson. So I have preferred to make a lesson of each step. So watch the next video for the